I'll be making this presentation on the work we've been doing on weeds and insects as part of the LIT project. When I say we, I'm referring to these four individuals who've done most of the work. Subodh Adhikari, a postdoc, Dane Elmquist and Jessica Kalin, entomology PhD students at UI, and Samuel Revelinsky, PhD student in weed ecology and genetics at WSU. And behind the scenes, you'll find Ian Burke and I, who are and me, who are supervising the work and mentoring the students. So why should we study these organisms? As you know, LIT seeks to understand the performance of alternative or aspirational farming systems in our region. Organisms like these can affect those systems and their viability, and they can change a lot depending on how we farm. So studying them should be part of LIT. But what questions to ask and how to address those? Here's the ones we're looking at. For insects and other arthropods, are there differences among the farming systems in soil surface or canopy dwelling insects, below ground arthropods within the soil, aphids and the viruses they carry? And some pictures on the right show some of these subjects. In weeds, how might important weeds respond to our management practices? And two are the focus of our work, downy brome and mayweed chamomile. And at the landscape scale, are there implications at that scale if alternative systems are adopted widely? And for that, we're focused on pollinators. So here's a sketch of our approach to answering these questions. First, for the pests and beneficial insects and arthropods. There we've been working primarily in the strip trials that you've heard about, where we've been doing sweepnet sampling in 2019 and pitfall sampling for 2 years to measure what's on the ground. We've also been looking at a seed removal rate experiment at St. John, and the reason for that is that ground-dwelling insects are mostly beneficials. They're either predators of pests, or they can take weed seeds to help manage weeds. So that's why we've focused on that one. And across the region, we'll continue to trap aphids in the fall and test fall-planted pea for virus. So pictured here is a pollinator trap used for some of the regional work, a pitfall trap that allows us to capture insects on the ground, a picture of the seed predation station that we've been using, one of the aphid traps here under autumn skies, and an array of Berlese funnels that extract insects from the soil so we can see what's down there. And here's just a close-up of the seed predation experiment where we have downy brome, mayweed chamomile, and lamb's quarter seeds all put out to see how fast they're removed. So for the pollinator work, a little more detail here. These are sites where Subodh Adhikari has been sampling pollinators associated with cover crops or with nearby wheat fields to get an idea of how those affect the overall abundance of these creatures. There are for the mayweed chamomile work, it's also landscape scale in its scope. So these are all sites, 11 sites at which mayweed chamomile has been studied. Subodh has been looking at the pollinators and other insects there and has collected seed from these sites, grown them in the greenhouse to compare their phenotypes, their germination, flowering time, plant biomass, etc., and study the degree to which that might be different genetically amongst the sites. And the other weed of concern is downy brome. Samuel is relatively new to the project. He's just getting started, but he will take advantage of the seed bank of downy brome, which allows us to see phenotypes of downy brome from some time ago when they entered the seed bank, and then look at how these traits in these in this downy brome change under when they mature under winter pea. Genetic studies of contemporary and seed banks plants can then allow us to understand and project how the weed could respond over a longer time to growing underneath winter pea. So what are some of our findings? So that's already been a lot of information. Now I'll apologize again because there's a lot of work going on and a lot of data. So I'm just going to share some highlights. Okay, for the crop canopy work, mostly the insect pests and predators that we find are not greatly affected by cropping system. Their abundance and diversity is similar to what we find across the inland Pacific Northwest in these crops. But second, though, on the soil surface, the communities of insects and spiders and other arthropods do differ noticeably. These pie charts are an example for 3 systems: soil critters under wheat, under winter pea, or under winter wheat with a cover crop. For example, under wheat with the cover crop, there are fewer ants, more rove beetles and ground beetles, who are predators, than under wheat only. Spiders are a bit more complicated because some spiders are less abundant and some types of spiders are more abundant under cover crops. For the seed removal experiment, rates of seed removal differ depending on the crop, and they also differ among the weed seed species that we tested: mayweed, chamomile, downy brome, and lamb's quarters. All of these tend to be removed more in fallow and less under winter pea. So what we plant affects this ecosystem service of seed removal by insects on the ground. No data on the viruses, except that here's a map of sites where we have measured the viruses on winter pea, and based on our data, there's not much infection, but winter pea can be infected before the onset of winter. So there's likely something to think about there if that were to become more prevalent in the future. Below ground arthropods are more abundant under winter pea at Genesee than the other under springweed or chickpea or winter wheat, quite a bit more. And in another trial done collaboratively with Michael Strickland and Bronte Sona at the University of Idaho, the types of below ground arthropods are more abundant. There's more different kinds under cover crops with a lot of species, a multi-species cover crop, than single species cover crops. So it's pretty clear that what we plant affects what's below the ground in terms of the arthropod communities, and those have implications for soil health because these animals are part of the whole soil system, and they are predators, and they also consume bacteria, consume fungi, they promote nutrient cycling and other soil processes. So they're part of the soil health picture that is apparently readily manageable and will be altered by future production systems. Now here's some of Sabode's work on the mayweed chamomile. Remember I told you that we collected seed from 11 sites and then grew plants from them to see if they differ. So what I'm showing here are plots of the flowering period. So you can see the flowering period in days of these greenhouse grown plants. One thing that happens is, one thing that's apparent is that at Cambridge, which is near Genesee, flowering period is longer than for plants from St. John. Climate and possibly management regimes seem to have selected for differences in this trait. Also, pretty important, among the progenies within site, so the mother plants there, flowering period differs significantly. Some flower for a much shorter time and others for a longer time, which means there's genetic variation that could respond to selection if we do other things on the ground as we manage or as climates change. So this particular trait is just one thing we've measured, flowering time, but it's important for the overall fitness of the weed because it requires pollinators, so flowering time is a constraint on its success. And another landscape scale study that Spo did, the cover crops harbor more bees, cover crops on the left here, and more kinds of bees than wheat systems. So as cover crops increase in the region, we can expect this effect to be evident across the landscape. A landscape that has somewhat reduced biodiversity now than it might have prior had to agriculture, and with more cover crops we can expect these things to probably have a landscape scale response. Oh, you might be wondering what Samuel found about downy brome. We haven't really done that yet. He's still working on it. So what's ahead? In 2020, we're going to continue documenting the effects of farming practices on the insects and arthropods, get a better understanding of that. We're going to determine the links, measure some of the links between the soil arthropod diversity that I showed you and soil health metrics. We're going to continue delineating the effects of cover crops on insect community regionally. This is focused on the pollinators. And we'll continue to monitor legume viruses in winter pea. Sabot is working now on screening mayweed populations for herbicide resistance because we know that these populations differ genetically. So it's important to find out if there are hotspots of herbicide resistance. And Samuel will use the downy brome genetics to assess how winter pea could affect the adaptive traits of that weed, especially flowering time, but other things. So we'll be busy. We have some questions for you. Are there additional pests, weeds, and diseases that you're concerned about in your aspirational production systems or imagined ones? And what questions or observations do you have about these and about the pests and weeds we're studying that should be addressed with future research? And finally, are you interested in the possibility of having the pests, weeds, and beneficials we're studying surveyed on your operation? Unless, of course, we're already doing that. And so for some of you, that's the case. So we'll see you and hope to have some discussion about these things on the 21st. And any additional questions you have for us, we'll see you on the 21st as well. So thanks for listening and take care.